All right, in this lesson we're going to look at meiosis, and before we do look at meiosis, there's something we need to clarify for you because this is one of those areas that can really trip students up, and that's the difference between an unreplicated chromosome and a replicated chromosome. The chromosome you're used to seeing is this guy right here, right, this one. This is the X-shaped chromosome that we looked at before. Remember, this is the original. Oops, that didn't go very well. Here. Original here. And then this side is the copy, right? All right, so this is what a chromosome looks like when it's replicated. In other words, this side of the chromosome here is the piece of DNA that existed prior to the S phase in interphase. Okay? I know that we don't have chromosomes at interphase, but if you looked at a chromosome during interphase, and if you looked at it during the G1, the chromosome during the G1 would look like this one that you see up here, because it's unreplicated. It hasn't gone through replication yet. During the S phase, this original piece gets copied, and the copy gets attached by the centromere to the original to make our X-shaped chromosome. So you don't really see these X-shaped replicated chromosomes until after the S phase in interphase, and they really don't appear until prophase, right? Because that's the first time the chromosomes actually start to appear. But if you could see a chromosome during the G1 or at the beginning of the S phase, what you would see is this kind of chromosome right here, which I call an L-shaped chromosome. Okay, an L-shaped chromosome as opposed to the X-shaped chromosome. The reason why this is important is because what you need to understand is there's going to become some distinctions that occur. When you have a chromosome that's replicated, even though you have half the number of chromosomes in a cell, you still have original information as well as copied information. So you still have twice the DNA in the cell, right? Because the copy and the original are together. Even though the number of chromosomes is going to be a half set, for example, and I'll show you what I mean in a few minutes. So understanding the difference between a replicated and an unreplicated chromosome is really super important. All right, so let's get into meiosis. Okay, now meiosis is about making the sex cells. We learned about that during the sexual and asexual reproduction videos. These terms that you see here that describe each of the phases ought to look familiar, right? This, what you're seeing in this picture, is meiosis 1. Meiosis 1. And what's important about meiosis 1 is you are separating the homologous chromosomes. Homologous chromosomes are paired chromosomes, okay? In other words, in each of these pairs that you see here, these paired chromosomes, one of those chromosomes originally came from the egg that made the cell, the other came from the sperm that made the cell, right? So one came from mom, one came from dad. Those are what homologous chromosomes are. And the homologous chromosomes contain the same genes, they just might contain alternate forms of those genes. All right, so the first part of meiosis is about separating the homologous chromosomes from one another. Now what's nice about this picture is up here at the top what you can see is what it actually looks like in cells when it's happening, and then down here it's showing you bigger an artist's drawing of what's happening. You'll also notice a lot of this stuff is going to look the same as it did in mitosis. There are a few specific things that change, but I'll point those out. So the first phase, meiosis 1, this is where you look slightly different from mitosis, and that's because of these homologous pairs. First of all, we start out diploid, okay? We start out in human beings with 46 chromosomes, right? That's what diploid means. You have 46 chromosomes. Now, remember During prophase 1, this is where crossing over occurs, and you can see the little crossovers there and there and there. Okay, so prophase 1 is where crossing over is going to occur. And that means you're going to get these mixed up genes. If you look, by the time you get to metaphase, look at the dark blue and the light blue. Little pieces of chromosomes are mixed up, right? So here's metaphase 1, and again, just like any other metaphase, everything is lined up at the middle. But notice also the big difference here is that at metaphase 1, you don't line these up in a row, you line them up in these homologous pairs. So the two big chromosomes are together, the two little chromosomes are together. Now they're only showing you 2 chromosomes at a time, or 4 total, so that you don't get confused. But remember, in human beings, we're talking about 23 pairs of chromosomes, right? 46 is 23 pairs. So you'd have 23 of these little pairs lined up in the center. Notice at anaphase, what you're doing is you're pulling those pairs apart. So when you end up at telophase 1, notice you still have these little X-shaped chromosomes. Now the question is, what's the chromosome number, right? Well, think about it. Back here at prophase, you had a total of 4 chromosomes, right? 4 chromosomes, 1, 2, 3, 4. You had 4 chromosomes here at metaphase, 1, 2, 3, 4. You had 4 chromosomes here. Now we have 2 cells each having 2 chromosomes, which means we've cut our number in half. By the end, you have 2 cells that are haploid at this point. So each cell is haploid. The thing is, because these are still replicated chromosomes, see how they're all X-shaped chromosomes? You still have duplicate DNA involved, and this is where I was telling you, yeah, you can be haploid in terms of number of chromosomes, but you still have duplicate DNA, duplicate information inside of the cells. So you're haploid, but you're haploid with extra DNA still. So we're not finished yet. We have to do something to separate out the original and copy from the chromosome. We have to make it so that all of our cells have unreplicated chromosomes. So we're going to do another division. All right, so what you're seeing now is the second division, okay? And notice we had 2 cells at the end, right? Each of our cells at the end was haploid, but each of our cells had the X-shaped chromosomes, right? The X-shaped chromosomes. We need to separate these X-shaped chromosomes so that they're unreplicated by the end of this, right? So notice again you've got metaphase 2, anaphase 2, telophase 2. So the names mimic mitosis. Meiosis 2 is really all about separating out the original and copied DNA, so the original and its copy from one another. So each of our cells has our 2 chromosomes that we started with. This time it looks just like mitosis, where now instead of pairs they line up in a row because you separated the pairs. So this time during anaphase 2 you're going to separate the sister chromatids just like you did in mitosis, and in the end you're going to end up with 4 cells. Each of them is haploid, but all of them have the L-shaped chromosomes, the ones that are unreplicated. Okay, so these are the L-shaped chromosomes, the unreplicated chromosomes. So now there's no copied DNA inside of our cells, okay? And that's what we were after. Unfortunately, we're still not done. We've done meiosis, so that's the process of meiosis. The problem is we haven't done gametogenesis, right? Remember, this process, these cells at the end of telophase 1 are supposed to be sperm or egg, right? We're supposed to be making sex cells here, and we haven't actually made the sex cells yet. So the next thing we have to do is gametogenesis. All right, so gametogenesis is going to look different depending on if you're in a man's body or a woman's body. So here is the male side, and let's go ahead and get this out of the way. This is the symbol for male. And you have to know that when we get to genetics. Okay? So that's the symbol for male. So what you're going to start off with is the cells that we ended up with at the end of meiosis, right? Or actually, these are the stem cells that are going to divide, okay, through meiosis. So here's meiosis right here. At the end of meiosis, you're going to end up with 4 cells. What happens is the nucleus kind of gets pushed to one corner of the cell. The rest of the cell gets dragged out. So see how the little sperm have these tiny little heads with a tiny little nucleus inside? The rest gets dragged out into a flagellum. So at the end in men, this process is going to result in 4 identical sperm cells, or 4 sperm cells. They're not really identical genetically, but they look identical on the surface. Okay. Now in men, this process goes from puberty until death. Okay, puberty till death. So gentlemen, once you start making sperm, you make sperm until the day that you die, which means that men are capable of actually having kids well into old age. Okay, and I think I have posted, if I haven't, I will post an Animoto about the difference between men and women in gametogenesis. Now let's go to the female side. All right, so here's the female side. Again, we start with a series. Oh, sorry. Let's go ahead and mark the female symbol. Here is the female symbol. Okay, that's the symbol for female. All right, so we've got these stem cells that will go through meiosis. But notice what happens here is 3 of the cells that we result from meiosis, right? 3 of them will surrender all their cytoplasm and all of their nuclei, or their organelles to the 4th one. Okay, so this little, when you get cytokinesis here, cytokinesis is uneven. This little cell ends up very, very small. This one ends up very, very large. When they divide, again, cytokinesis is uneven here. So you get a lot of cytoplasm and a lot of organelles. The rest of these 3 kind of die off. These are what we call polar bodies. These 3 cells are polar bodies. This becomes the egg that's released. Okay, so in females, instead of 4 cells, you end up with 1 cell. Okay, so males get 4 sperm from this process. Females get 1 egg cell released. This process, the difference is that this happens before birth. Okay, in fact, the before birth part is right there. Okay, so everything from here on up has happened before birth. And then what happens is once a month, a woman's body will go through the rest of meiosis and release 1 egg. And that release of the egg starts at puberty, and then that ends at menopause. Okay, so this doesn't happen throughout a woman's life. Okay, so before a baby girl is born, she's produced all these little pre-eggs that she's made. And then once a month, once she hits puberty, she starts releasing. She'll finish out meiosis and release 1 egg per month.